Okay, we got the signal to start the festivities. It's a great pleasure to welcome Gail Mandel here today for the Storer lecture. And Gail, I'll give you some nuts and bolts at the beginning here. Gail did her bachelor's degree and her PhD at UCLA, and her PhD research was with Bill Wickner on bacteriophage M13 coat protein synthesis, so she was a real membrane protein biochemist. And then went to work with Mel Simon when he was still at UCSD on phase transition in bacteria, and did a number of nice papers on that topic before moving as an instructor to Harvard Medical School in the pathology department, where she worked with Thomas Benjamin on polyomavirus. And at that point, her and her husband Paul stayed in Boston, and Gail took a faculty position at Tufts Medical School, New England Medical Center, and worked in this newly created Department of Molecular Medicine with Dick Goodman, and worked on cloning, neuropeptides, VIP, somatostatin, others, and actually was also involved in the instrumental work that led to the definition of the cyclic AMP response element. And then ultimately the CREB protein that binds there, and then everybody knows about CREB now. But Gail was really involved in some of that early work that laid the ground for all that later important identification of those proteins. In 1989, she moved as an associate professor to SUNY at Stony Brook, and then was there for 15 years, I guess.
Almost. Yeah.
Yeah, somewhere around there, and
17,
rose to full professor, and in 2007 moved up to Portland to the Volum Institute, which is associated with the Oregon Health and Sciences University, as I was instructed to say by faculty colleagues up there, and she's a senior investigator there. Gail's also a Howard Hughes investigator, and she got this in 1997 when she was at Stony Brook, and she was and remained for the whole time she was at Stony Brook the only Hughes investigator in the entire SUNY system, which I looked up this morning because I knew I had heard at one point that the SUNY system was the largest university system in the world, and I verified that this morning, and there's 85,000 faculty members in the SUNY system, and Gail was the one. And now they have zero, now that she's gone.
And they don't care.
that had a Howard Hughes investigatorship. And you know, the thing is, right, you would think that a exceptional honor like that would be something that might go to someone's head or cause them to become, you know, a different person or not. But, you know, Gail, at that first moment and forevermore, I mean, she knew she was taking on this huge responsibility, and there was going to be a lot of pressure. But from the get-go, it was like, how can I use this to help us, to help my colleagues, to help us? How can I leverage this to get more resources from the university for basic research? And it was really, you know, impressive that to see someone that I had known for a while respond in that way. And, you know, I mean, it was really a very good indication of what Gail's all about. So what she's going to talk about today is rest and regulating neuronal specific gene expression. She's going to give you a little bit of history, but I think what she's not going to say, she's going to give you the history on how she came to identify rest through working on sodium channels. She's probably not going to say that her original cloning of the sodium channels basically broke open an impediment for ion channel research that had existed for a number of years, because the original ion channels that were cloned by a single lab in Japan who refused to give out any clones to anyone. So there was this kind of gap in the field of, okay, we have the sequence information, but nobody can really act on it because there's no cDNAs available. So Gail took it upon herself to, you know, single-handedly clone out sodium channels and basically seed the entire research community with her generosity of these clones. So everything we know about voltage-gated sodium channels, their mutations in neurological disorders, the hyperkalemic periodic paralysis tests that's done on quarter horses at the vet school here, is all done using clones that were derived from Gail's original cDNAs that she sent out, and which were the springboard for the identification of RES, which was Gail's real interest, but basically helped the entire field of ion channel biophysics and ion channel research by doing this. So I don't have the seminar title, but it's something about a series of rest stops on the way to neuronal differentiation. So thank you very much, Gail, for coming, and we look forward to your talk.
Thanks. Yeah, you do deserve an applause for that introduction. It was really, really very generous, and I appreciate it. It's terrific to be here. I grew up in LA. As Jim said, I'm a graduate. I did my undergraduate at UCLA and a postdoc at UCLA. And it's true what they say, you can take the girl out of California, but you can't take the California out of the girl, and I really love being back. And it's also been a treat because I have several old friends here. One, of course, is Jim Trimmer. Another is Don Beers. I haven't seen Don in 30 years, but we played on the same softball team at UCLA. I, of course, was the pitcher. And I don't remember what position that you played. I'm sure it was memorable at the time, but I think you were a long ball hitter, if I remember correctly. And so it's been 30 years, so that was pretty amazing. And I also have other connections with people here. For example, with Janine, we have a mutual interest in Rett syndrome, which is something I'm not going to talk about today. And of course, I've been a long admirer of Peggy's work, and we had a really great discussion. And I also learned a lot about other aspects of gene regulation that I'm very interested in. So it's just been a terrific morning. And I'm really grateful for the opportunity to present my work today, in part because a great deal of what I'm going to talk about today is something we haven't published, and so I'm kind of trying it out on you guys to see how it goes. So, as Jim said, I've had a long-standing interest in control of the neuronal phenotype. And our original idea for how to study the importance of gene regulation in controlling neuronal phenotype was to pick a feature of a neuron that was essential for its function and then work backward from that protein to its gene regulation to try to understand what it was that allowed this trait to be specific for neurons. And of course, the trait that really comes to mind, especially if you're my generation, in terms of making the nervous system special, is the ability to generate action potentials. And this is an action potential that was taken from a classic study by Hodgkin and Huxley, in which they were monitoring the generation and propagation of action potentials along the squid giant axon, so classical electrophysiological preparation. And the action potential looks the same in a squid giant axon and any other excitable cell. So it's a very highly conserved signaling mechanism. And the upswing of the action potential is due to the rapid opening of voltage-dependent sodium channels in the membrane of the excitable cells. So for me, it was a clear hallmark property that I reasoned would provide insight ultimately into the control of neuronal phenotype. Now, as Jin generously mentioned, my lab, as well as the late Dr. Numa's lab, were very interested in applying which was then newfangled molecular cloning techniques to determining the primary structure of this very important ion channel. And the deduced primary structure is shown above. And I should say that during the course of our cloning, we determined that sodium channels were encoded by a multi-gene family. There's maybe 12 different genes that are closely related but functionally distinct. And this is a predicted primary structure. It's one of the few voltage-dependent channels for which there's no yet crystal structure. And I also want to say that the cloning of the sodium channel was done together with Jim Trimmer. In fact, Jim lived at our house for a while, got to know each other very intimately. During the time we decided to try to clone the full-length sequence for the skeletal muscle sodium channel. So, of course, the other excitable tissue compared to the nerve is skeletal muscle, and we were successful in that. And then actually Jim went on to study more of the biology of this ion channel in very elegant ways. The bottom part of this slide just points out what I said, which is that the sodium channel is encoded by a large multi-gene family, and the genes tend to be clustered in different chromosomes. And the asterisks indicate specific sodium channel types that have been implicated in human disease. So the sodium channels are another class of ion channels that have been a target for a lot of human diseases. And it also explains why patients that have hyperkalemic periodic paralysis, which is a skeletal muscle defect, have perfectly good cognitive abilities because the brain sodium channels are intact. So we made probes from the cDNA for brain sodium channels and then convinced ourselves that the expression would be relegated just to the developing nervous system. And we did this by in situ hybridization. So this is a picture of an embryonic day 10.5 mouse embryo. And the purple here represents a brain sodium channel transcript. This actually is trapping, a little bit of trapping of the probe. So this is really kind of an artifact. And if I waited a little bit longer in time and showed you an in situ picture, what you'd see are some purple transcripts along the spinal cord. But importantly, this particular sodium channel type is never expressed outside of the nervous system, so it truly is a neuronal specific channel. And our hypothesis was that the genetic mechanisms that restricted the sodium channel to the nervous system would also be restricted to the nervous system. That is, we were envisioning a sort of positive regulatory mechanism that worked together in order to give neuronal specific expression. Why we thought that was because investigators studying tissue-specific expression of other tissues like liver, kidney, muscle had found that the expression of those cell-specific proteins was dependent on a network of positively acting transcription factors. So we thought the same should be true for the nervous system. But that hypothesis turned out to be entirely wrong. It turned out that the mechanism that restricted the sodium channel to the nervous system was a repressor mechanism. And I've shown the basic components in this slide. So just imagine this is the cartoon of the sodium channel gene. About 1,000 base pairs upstream from the promoter of the sodium channel gene, we identified a very large site, repressor element, that we called RE1 for repressor element 1. And there was nothing very, there were no clues from the sequence that would tell us anything about how this mechanism worked. But one interesting thing was that David Anderson at Caltech was, at the same time that we were studying the sodium channel, he was studying another neuronal specific gene called SCG10, which is a phosphoprotein which is found in synaptic terminals. And this was, of course, before databases. And one day at a meeting, David and I sat down and noticed that the elements that conferred neural specificity of these two genes were both highly conserved. And so this then led to the idea that any insights we were going to glean from studying the repressor mechanism or the neuronal specificity of the sodium channel was going to have wider implications for control of neuronal phenotype in general. So this is only half the equation then. The other half is the protein that recognizes this repressor element. And we cloned the cDNA for this protein, and we called it REST for RE1 silencing transcription factor. It turned out to be a zinc finger protein with 9 zinc fingers, 8 of which we thought are somehow associated with binding to this element, and then a third one with a slightly different property. Now, if this model is correct, then REST should be expressed reciprocally to the sodium channel gene. That is, wherever there's a neuronal gene on, there shouldn't be any rest, and vice versa. And so, before I show, well, I can show you that next. So we tested that again using in situ hybridization. This is the picture that I showed you before, and you can see there's very nice reciprocity between the expression of the REST repressor protein and the sodium channel, and in fact, where the nervous system is going to develop. Recently, we used a sequence analysis approach to really get a better handle on the extent to which REST might regulate neuronal phenotype. How many genes are under REST control? Is it really a master regulator? And I think it really is. So in the course of doing a sequencing analysis, that is trying to identify all the REST binding sites in the mouse genome, we uncovered another site that was slightly a variant of the site I showed you previously. And when we add up all the sites together, there are approximately 8,000 to 9,000 genes which contain REST binding sites. And of course, we haven't analyzed every single gene, but all the ones that we've analyzed are either expressed exclusively in the nervous system or in neurons and some other tissue. And the targets for the REST protein encompass all the different aspects of neuronal phenotype. So many of them are involved in synaptic signaling, growth factor receptors, growth factors, big families of ion channels and transporters, big families of adhesion molecules, some transcription factors, molecules involved in axon guidance, and so on. And it's really kind of interesting to me that in these big gene families that have obviously undergone gene duplication, you might have thought, particularly in the case of the olfactory receptor genes, that there'd just be a single REST binding site that would somehow control these clusters, because these genes are all kind of clustered on the chromosome. But in fact, it seems that most of the individual family members have their own REST binding site. In the olfactory system, what it may do is provide a sort of negative platform where all the olfactory receptors off. You may know that an individual neuron only expresses one olfactory receptor type. And so maybe through evolution, it's just easier to start with everybody being off and then sort of plucking out the one you need when you need it. And actually, there's a group at UCSF that's currently testing this idea. So this picture of the reciprocal distribution of REST and its target genes has led to several questions that we've been trying to address over the last few years in the lab. And one of the questions is what the role of REST is in vivo. Another question is, what is the mechanism by which REST represses all its different genes? In particular, for example, what is the mechanism by which REST can exert long-term transcriptional repression throughout the life of an animal to keep neuronal genes off? And what's going on in the nervous system? Is REST never expressed in the nervous system, or is REST expressed in progenitors and then down-regulated as differentiation proceeds? And if that's the case, then how is REST controlling neuronal differentiation? So I'm going to give you some representative experiments that address those questions. First thing I want to say is that REST doesn't work by itself. And early on, we identified, using a combination of two hybrid and biochemical analyses, two co-repressors that are recruited by REST to neuronal genes through this REST binding site. One co-repressor is one that many of you may be familiar with, called SYN3A, that's in a very tight complex with histone deacetylases, which obviously can modify the chromatin. This complex had been previously identified by investigators studying steroid hormone receptors. And this is a repressor complex which can either be a repressor or an activator. So steroid hormone receptors can either be repressors or activators depending on whether or not ligand is present. And this one didn't seem like a really good candidate for long-term repression of neuronal genes. So we hypothesized there must be another one. And in fact, there is, and we call it co-REST. So there are two repressor domains in REST, and each one of them recruits a different co-repressor complex. And this one also, co-REST, also recruits histone deacetylases. But as I'll show you later, this is kind of just a core complex, and it does raise, because of the presence of histone deacetylases, it does raise the question of the relationship between REST repression and chromatin modifications, and I'll be talking to you a little bit about that in a few minutes. So with respect to whether or not REST might play a role in the nervous system, we've examined this in a couple of different ways. And one of the simplest ways was to examine the role of REST during the differentiation of embryonic stem cells through neuroprogenitor stage to non-dividing mature neurons. And the reason this system was appealing to us was because it's very amenable to doing the kinds of molecular and biochemical studies that we wanted to do, not just to study how REST causes maturation of neurons, but also to understand the relationship if there was one, between rest and chromatin around the rest binding site. So the first question is rest expressed in embryonic stem cells? And all the experiments I'm going to show you have been done with mouse, for those of you who study embryonic stem cells. And the answer is yes. In fact, it's expressed to really high levels in embryonic stem cells, along with many other proteins that are expressed to high levels. So this is an immunochemistry picture of the nuclei of mouse embryonic stem cells using an antibody that we made to the rest protein. And here is a Western blot showing high levels of rest protein in ES cells compared to fibroblasts. So fibroblasts don't express any neuronal genes. They certainly don't express the sodium channel gene. And there is rest, but you can see that the level is much lower than in the embryonic stem cells. And I'll come back to that in a few minutes. So rest is there. And then the question is, do you need it there for anything? And so we've tried to, or maybe more specifically, rest is there, but is it acting as a repressor on these genes? Because just because you see something by Western doesn't mean that it's actually a functional protein. So we've addressed this question in a couple of different ways. And one way is to make a lentivirus that transduces an shRNA for rest. And we can infect ES cells with this transducing virus, and you can see that we get a very nice knockdown of the rest protein in the ES cells. And we see significant decrease in the RNA as well. And, oh, I thought it was just a Mac thing, but it's okay. So then the next question is our shRNA for rest? We don't see anything by Western, but are we really removing it from the chromatin? This is actually, I talked to some people about this morning. This is actually kind of problematic for many transcription factors. You can knock them down and use the criteria of Western to say that they're gone, but when you actually look at the chromatin, you can often find it still there, and that does seem to be the case for rest. So what I've shown you here is a chromatin immunoprecipitation for rest on one of the rest target genes, in this case synaptophysin, which is a classic neuronal protein. And what we've done is made sort of a mini tiling using primers that surround the rest binding site, which is shown by this blue vertical arrow here with respect to the synaptophysin gene, which is shown here in blue. And then we monitored our chromatin immunoprecipitation using these primers. And you can see we get a nice peak very close to the rest binding site. This is sort of characteristic for the kind of pattern that we see. And then when we do the shRNA, which you see in blue, we do reduce the amount of rest that's associated with synaptophysin, but it's clearly not zero. And so we have to take that into account in interpreting some of our experiments. For some experiments, it doesn't matter that we don't get a complete knockdown. For some, it does, depending on what conclusion we wanted to draw. But in any event, this did tell us that rest was not just present in ES cells, but was actually associated with the promoter of neuronal genes. And then the question is, if it's acting as a repressor and you remove it by knockdown, do you see any derepression of neuronal genes to say that it's functional? And in fact, you do. So this is a qPCR analysis of some of the rest target genes with some controls. And you can see that there's actually a broad range of derepression of these genes. And so this is kind of a place where not having a clean knockdown is kind of a problem, because we don't really know whether this range is due to the fact that there's a different genetic context to where rest is binding. So you remove rest from some genes, and maybe there's another repressor there, and so you don't see a big derepression. Or is it that for particular genes we haven't released all of the rest, and so there's more or less rest there? So I can't tell you that. I can only say that there are some genes, like synaptophysin, where we don't get complete removal of rest, but you can see there's a very large, almost 70-fold derepression of the synaptophysin gene. So this is clearly a gene where rest plays a predominant role. In fact, we know there are different sets of neuronal genes. Some genes lose rest and then pop to a very high basal level by default. This is normally during normal neuronal differentiation. And some lose rest, and they pop to a very low basal level of expression. And that is likely due to a different context of the factors that are there. But in any event, rest is present in ES cells. It's bound to genes that it's supposed to be bound to, and when you remove it, many genes are de-repressed. So it's certainly a functional protein in ES cells. I'm going to talk about the biology of what happens when you do this, or the lack of biology when this happens, a little bit later in the talk. Okay, so rest is present as early as the ES cell stage. What about the neural progenitor stage? Is rest still there, or is it gone, or what's going on? And so this is showing you an in situ hybridization picture again of rest in the ventricular zone of a developing mouse, and the blue transcripts here are monitoring the presence of rest in ventricular zone cells. So this is the portion of the developing brain which is very active mitotically, and that's kind of where you would expect it to be. But the funny thing was, when we did a Western blot, we couldn't see it. So below here is a Western. This is the ESL signal that I showed you before, which is quite robust. If you look at E12.5 cortical progenitors, which we isolate acutely in culture, we expected to see rest protein, which would sort of be equivalent to the RNA we saw here, but we saw nothing. And so because we saw RNA but no protein, we thought the most parsimonious explanation was that at this particular stage in development, rest was starting to be turned over and being targeted to a proteasome pathway, as if you want to be anthropomorphic, as if the progenitors are trying to get rid of all the rest so that they can be relieved and be expressed to form a neuron. So we tested that by treating these cortical progenitors with MG132, which is an inhibitor of the proteasome pathway. And you can see when we do that, we can rescue rest protein. So that's kind of compatible with our idea that rest is turning over. And in fact, our lab and several other labs are now trying to get at the mechanism by which rest is being targeted to the proteasome pathway. Well, what about rest at this particular time? There's just a little bit of rest left, and it turns out that that rest is clinging dearly to the rest binding sites of these neuronal genes. So the cell's getting rid of 90% of the rest, but the rest that's left is still on the chromatin, and it's still acting as a repressor, because when we introduce, in this case, an adenovirus, which is expressing a dominant negative form of rest, we can de-repress some rest target genes. So rest is at much lower levels in progenitor cells, but it's still there, and it's still repressing, presumably keeping neurons from terminally differentiating. Okay, what about immature neurons? This has been a very controversial issue in different labs that are studying the role of rest, and I can only say that in our hands, with our reagents and our cells, we don't detect rest in post-mitotic mature neurons. And so that's shown here by this immunochemistry picture. This is what I showed you before, and we just don't see it. We don't see it by Western either. So we think that somewhere between here and here, rest is disappearing, and we can't rescue it in cortical neurons, in this case, by treating with MG132. So that suggests it's not just because it's really getting turned over. It's more likely that rest itself is being repressed at that stage. And we've been able to identify, just look at the right panel here, we've been able to identify a repressor complex that is in fact bound to the rest gene close to the promoter. And this repressor complex contains the retinoic acid receptor, so may be mediated somehow by retinoic acid in a way that we don't understand yet. So this slide shows that rest and its companion proteins that I showed you earlier are present on, in this case, the synaptotagmin 4 gene. This is a chromatin immunoprecipitation. And there's a DNA binding protein called methyl CpG binding protein 2, which is the gene that's defective in Rett syndrome, but I'm not going to talk about that today. That's also present with REST on this gene. In cortical neurons, I showed you REST isn't there. Presumably because REST isn't there, these other co-repressors are no longer being targeted to the REST gene. And this slide just shows you that the expression pattern of REST target genes, neuronal genes, coincides very nicely with the disappearance of REST. So this is a summary of what I've told you. In stem cells, there's a very high level of REST expression. REST is collaborating with some other components on REST target genes. These genes are repressed. In progenitors, REST starts being turned over by a signal that we don't yet fully understand, but the end result is that most of the REST is degraded, leaving a little bit on progenitor DNA. And then around the time of the exit from the cell cycle, REST is itself transcriptionally repressed, and the neuron is free to then elaborate a fully mature phenotype. So now I want to talk a little bit about the mechanism by which REST is orchestrating, or is it orchestrating this repressor mechanism. So the data that I've showed you is very correlative. So what I want to talk about in the rest of the talk is evidence that we have that it really does matter whether REST is there, and also how REST is affecting its repression. Now, I know many of you in this audience, being in the Genome Center, being experts on this, appreciate the fact that there are nucleosome structures in the chromatin with DNA wrapped around histone cores, and that the tails of the histones can be modified post-translationally. And once they're modified, like any other protein, they act as docking sites for other proteins. And there's a wealth of different modifications that have been described, and I'm mainly going to talk about a couple of them. One is methylation of lysine 4 on histone 3, for which there's a lot of evidence that trimethylation at this site is associated with transcriptional activation. And conversely, methylation of lysine 27, which is mediated by the polycomb complex, has been almost exclusively associated with repression or silencing. There are other modifications, like dimethylK9, for example, is one that's been associated with repression, but trimethylK9 can be repression or activation. It's a little less clear. Acetylation of 9 and 14 is often associated with activation. But for me, rather than studying every single modification, we've sort of focused on K4, K9, and K27. So we first started looking at the complex that REST uses to repress in non-neuronal cells that are terminally differentiated, like fibroblasts, or kidney cells. And I showed you this complex before. The thing is, though, in a fibroblast, if you treat that cell with something that abrogates histone deacetylase activity, like trichostatin A, you don't see very much derepression of neuronal genes in those cells. And so that is kind of what suggested to us that REST might be working with histone modifications to pull in other enzymatic activities that would be helping REST repress. And so we characterized the chromatin landscape around REST binding sites. And what we found was that often the REST binding site was associated with methylation of the DNA, which of course is a different kind of epigenetic modification. And there was a lot of dimethylK9, and there wasn't any K4. Now, at the time, actually, we did these experiments, there weren't any good antibodies for K27, so we hadn't interrogated it yet. And then, in addition, we found the presence of DNMT1, and again, this methyl CpG binding protein, MECP2. But this is a picture that looks very reminiscent of a true silencing complex, which is extremely compatible with the fact that we don't see any neuronal gene expression in fibroblasts. So this was kind of satisfying. However, this kind of architecture is not very compatible with another type of non-neuronal cell, namely ES cells, which want to go ahead and differentiate into a neuron, because this is kind of what you would imagine as a permanent repressed structure. That's not going to be so good for an ES cell or a neural progenitor that's ultimately going to want to express a neuronal gene. So we hypothesized that rest must be associated with a different kind of architecture in different non-neuronal cells, in particular stem cells. And sort of as a corollary of that, we monitored by RT-PCR expression of two neuronal genes, in this case calbindin and synaptotagmin 4, in ES cells and fibroblasts. You could see even at high cycle PCR, we don't see any RNA from these genes in fibroblasts, but you do see a little bit of transcription of RNA in ES cells. And as I told you before, if you treat with fibroblasts with trichostatin A, you don't see any derepression. But if you rattle the cage of an ES cell, you do now start to see some repression. So this is all pointing to the direction that the landscape of the chromatin is very different for neuronal genes in ES cells than it is in terminally differentiated fibroblasts. So we sort of thought, well, maybe the complex looks different. Let's just look at the basic proteins that are present. And when we did that by chromatin immunoprecipitation, actually the basic components look the same on these two genes in these two different cell types. So we knew there had to be more. And in fact, the landscape looks really pretty different. So this is the picture I showed you before, and this is the picture in an embryonic stem cell. You can see the core is the same, but there's no methylation of the DNA associated with the rest binding site in an embryonic stem cell. And furthermore, turns out there is a little bit of dimethyl K9, but nothing like what you see in a fibroblast. So this is that repressor mark. There was di and trimethyl lysine 4, which is consistent with activation, but very low levels. And a little bit hard to understand, and I'm going to get back to this, but basically dimethyl K4 was all over the map, seemed to be very gene dependent. Trimethyl K4 was low uniformly. So there were some hints that this should be, despite the fact that there was a repressor complex sitting on this DNA, there were some hints that there was something funny going on, because instead of seeing repressive marks, we saw activation marks. And consistent with the fact that I told you that we could see RNA, we could also see Pol2, RNA polymerase II, associated with this. So very, very different architectures for these two types of cells, but both of them had the same core rest complex. So we were interested in trying to figure out what was going on. One thing I want to point out, I showed you this before, I told you that the rest gene is expressed to really high levels in ES cells, and there's a lot of rest protein. That may be due to the fact that three factors associated with pluripotency, NANOG, OCT4, and SOX2, are associated with the rest gene. This was actually shown by Rick Young and his colleagues at MIT. And it's likely, I think, although we haven't tested this directly, that this triumvirate is acting as an activator complex on the rest gene, which is causing its expression level to be so high. Well, then this poses a little bit of a dilemma, and I'm not going to be able to answer this dilemma today, but the problem intellectually is that in a fibroblast, you have really relatively very little rest protein, but these genes are silenced. In ES cells, you have 10 times the amount of rest protein, and these genes are only repressed, transcriptionally repressed. And so what's going on there? One thing that this picture suggested to us, we call this in the lab the ESL Dilemma, is that there's got to be more to the story. And so we decided it would behoove us to try to find out more about the rest complex. And so we've done this in collaboration with John Yates' lab at Scripps. The idea was to generate an embryonic stem cell line which expressed pretty much physiological levels of rest with a tag on it, which allowed the transfer of biotin in vivo to this protein. And this embryonic stem cell line also contains the E. coli BRA enzyme, which transfers the biotin to the rest protein. We actually got the ES line from Stu Orkin, who was kind enough to supply it for us. And then we just do an IP. The advantages for using this particular approach are shown here. And then we sent it to John for analysis of the complex. And I just show this to give you a little bit of an idea of what the raw data looks like. So because it sounds so simple, you just send your sample away and you get a list of proteins and you figure out which one you want to study. Sure. So just to give you an idea, so this Venn diagram shows, the idea is to do several completely biological independent experiments and do the pull down separately in those three. And so this Venn diagram shows you that you don't get a hell of a lot of overlap. Okay? And then you get a list of proteins that have been ID'd, and then you can decide what criteria you want to invoke for saying that an associated protein is really associated. So this table shows you a very stringent criterion, which is that the proteins had to be present in all three pull downs. And I'm going to tell you that is really pretty stringent. And in most cases, we asked, not all, but in most cases, we asked that it not be present in the control pull down. Actually, I see that this table is kind of a composite of a couple of different criteria. But anyway, what was satisfying is that we did identify proteins that we knew from our experiments and from several other independent labs were associated with rest. For example, co-rest. Okay? Everybody knows that rest is associated with co-rest, and you can see that there's very nice, well, first of all, it was satisfying to see that we found rest. That's always kind of nice. And, but then we did find co-rest, and what was interesting is we knew there were 3 different isoforms of co-rest that are similar but different, and it looks like co-rest is co-rest 2. Maybe they're forming some kind of dimer, which is part of the complex, and we don't exactly know why. Oh, the other criteria you can look at is the peptide representation. You know, how many peptides of that protein? Look at rest, 41 peptides. You get really good coverage. But as you work your way out from the DNA binding protein to the peripheral interactions, you're going to get weaker and weaker interactions. And we only do the pull downs so far at 1 salt concentration. So, you know, there's kind of a lot of things you have to consider in figuring out what's real and what's not. Syn3A we knew, and there's very nice representation there. The histone deacetylases, they're there. That was nice. Actually, everything in yellow we knew should be there. So an interesting one is LSD1, which is a lysine demethylase. And that was a little perplexing for us because we didn't expect to see any lysine for methylation because of the presence of this demethylase, but in fact we do. And it turns out that if you remove, well, I'll talk about this later. Actually, it turns out to make sense in the end. And then G9A is a histone methyltransferase that we and several other labs showed was there, and that was there. In blue are new proteins with new activity, some of which we're interested in following up and some not. So one thing that was interesting is there were several chromatin remodelers. And I think that makes sense when you consider the fact that you have a repressor sitting on a promoter that's transcribing. So it sort of makes sense in terms of the chromatin being opening, and also kind of what we think about ES cells in terms of the chromatin being accessible. There's also an adapter which we now know links rest to the histone methyltransferase. So most of the proteins that we got from the complex make sense, and then there's a long list of things that don't. So this is kind of our newer picture of what the rest complex is in embryonic stem cells. And I've put some of the new proteins in context here. There are a couple of other interesting things. One is I told you there is some lysine methylation despite the fact that LSD1 is here. And actually the reason is because if you take LSD1 away, the methylation goes way up. So in fact, what LSD1 is doing is keeping this methylation at a low level. It's not that it disappears entirely. One of the surprising things is that we found the presence of methylation of lysine 27. This is the modification that's catalyzed by polycomb protein. And this was surprising because we didn't see initially any polycomb proteins in our complex. And so this link was kind of a mystery. In addition to the proteins we expected, there are a couple of classes of proteins which are quite interesting. There's another class of enzymes, one of which is an arginine methyltransferase, which hasn't been described as being associated with repression. So we're interested in following that up. And there are also a couple of tumor suppressors which have popped up in the complex. And that's interesting because outside of the nervous system, Steve Elledge's group has published that REST can be a tumor suppressor. And so this may help us understand what that link is. So I showed you this before. This is just reminding you that we do see derepression if we remove REST, and that's important because now we want to ask how the chromatin modifications change if we remove REST. And I think this is really an important experiment because with all the global experiments being done now, all the global chromatin immunoprecipitations, which are looking at marks genome-wide, I think to know how those marks actually are interpreted in terms of function, you have to start removing, I think, the factors and asking how those marks change. And so we have an advantage that we can do that for REST because we know all the target genes and we know where all the REST sites are. So this experiment is, again, chromatin immunoprecipitation. This is the same synaptophysin gene that I showed you before. And now we're looking at modification of the lysine 4 mark. And I told you that if you remove LSD1, it goes down, and it goes up. And if you remove REST, you see the same thing. You see a nice peak of lysine 4. So this is saying that if REST wasn't there, these neuronal genes, I think, would be expressed at a pretty high level. And what REST is doing is suppressing the level in part by collaborating to constitutively remove this lysine activating mark. We were very curious to see what we'd see for lysine 27 because I told you that we didn't have a link to polycomb. And we saw a very robust removal of lysine 27, methylation of lysine 27 when we removed REST. So that again said that that landscape, it wasn't just a correlation between REST and that landscape, it was actually a functional relationship. So this is our model. Our current model for our thinking for what's going on. In wild-type embryonic stem cells, we are imagining a REST complex that has the components that I showed you. And when we went back to our mass spec analysis and looked at our list carefully, we were really pleased to find one of the subunits of the polycomb complex. And that subunit is SUS12. And we're hoping Peggy will give us some antibodies so that we can confirm this. But so we're sort of hypothesizing that the link between REST and the repressive modification on lysine 27 is going to be through SUS12. So SUS12 is one of the triumvirate of proteins that makes up a functional enzyme which does the methylation of lysine 27. Now here's the interesting thing. If you take REST away, okay, no trimethylation, that's fine. We see increased neuronal gene expression. That's fine. But in fact, there's absolutely no effect on the biology as far as we can tell. So despite the fact that you remove the repressor, you remove the chromatin marks, you get upregulation of the genes, we can't see any effect on the self-renewal of embryonic stem cells, nor do we see any effect on the ability of stem cells to form a neural progenitor. So we interpret this data to mean that it's really hard to interfere with the self-renewal property of an embryonic stem cell. So we're sort of the mind that stem cells are really committed to dividing, and that they don't have the other factors that you need just in an ES cell to make a different lineage. So you can't just simply upregulate a marker for a lineage and get that lineage. And that's kind of maybe how the ES cell protects its ability to self-renew. We're not the only ones who think this way. Terry Magnuson at UNC also has data on coming from a completely different perspective, which I think provides even better support for that idea. So what we want to do to finish this study is to examine neuronal genes in ES cells that are missing some of the polycomb components. So we sort of want to do the reciprocal experiment. And there we'll look at trimethylation of K27. I'm anticipating we're going to see increased neural gene expression, just like when we removed RES. And I'm also anticipating that there will be no effect on the ability to form a progenitor in those cells, but we'll see. So in the last few minutes now, so what I'm saying, I guess, is that that rest is here, and it's here, although at much reduced levels. But removing it at this point doesn't have much of an effect, both because of the biology of the cell it's in, but also, I think, because of the lack of the factors that you need at that stage to work with rest in order to give a neuron. What I'm saying is removal of rest is not sufficient to give you a neuron at the wrong time. It's not that kind of a master regulator. So then the question is, how important is rest between this stage and exit from the cell cycle to become a mature neuron? And this is actually the stage that I think rest is most important. And so in the last few minutes, I'll just tell you some of the in vivo experiments we're doing to sort of test this. So one type of experiment we can do is a gain-of-function experiment, where we can overexpress rest in progenitors during differentiation and ask whether that has an effect. And this is an example of an experiment where we test the idea, and we use a technique called in utero electroporation, where we can inject expression vectors containing rest or containing rest plus RFP or GFP into developing embryos into the ventricle, and then apply a current and sort of send the DNA in the direction that we want to send it. So in this case, this is actually done, we did this experiment in rat, and we did the in utero injections at E14 and then sacrificed the animals at E16.5 and asked what the disposition was. And so the injection site, the ventricles are here in the bottom left. And so if you have a control vector that just expresses GFP and RFP, you can see that the cells, and you're hitting cells at E14. These are nestin-positive neural progenitors, and those are the only cells that are taking up the DNA. And you can see that you get very nice migration of those progenitors through the cortex, through the cortical plate, where they presumably are differentiating and stopping to divide. However, if you have rest on that vector, and in this case, we're swamping out the proteasome pathway, so we're overexpressing it and keeping it persistent. What happens is that these progenitor cells, they get stuck. And they get stuck in migration and differentiation, and they're stuck in between the ventricular zone and the intermediate zone, which you can see by this green color, and the quantitation is below. So between the progenitor stage and mature neuron stage, that stage seems to be sensitive to rest by gain-of-function type of experiments. And what we're doing now is culturing these cells and figuring out what their markers are to see exactly what stage of differentiation they're stalled in. And actually, it's a little bit past the nestin stage and a little bit before the mature neuron stage. So it's some kind of intermediary stage. Okay, what about the other type of experiment? What about knocking rest out conditionally and asking what the effect is? So this has been a very difficult problem. And it's been difficult because the gene structure is not user-friendly for rest. So if you try to remove the amino terminal exon, coding exons, you don't put the rest of the protein out of frame. And there are many ATGs in all frames, and they're used. So if you take your targeting construct and you transfect into cells, you will find rest protein. And since there are functional domains of rest throughout the protein, it's kind of a mess, so you don't get a clean result. So we decided to take another approach. And this really came from me being bored one day and surfing the net and ending up in Germany somehow at some GGTC consortium where there are a group of investigators who want to knock out every gene in the genome using a gene trap methodology. And so the idea is to capture a gene trap that contains the LAC-Z gene in a transcriptional termination site into the first intron of your favorite gene. And then what happens is you splice the first exon to this exon, and there's a transcriptional termination, so you get LAC-Z, but you don't get the rest of the protein, so you have a mutated protein. And it's designed in such a way that you can flip this cassette to render back the wild-type allele, and then you can flip it again with Cre recombinase and generate your mutation, just like you did to begin with. Now, this is a very idiosyncratic technique in the sense that some genes may not like this cassette in the first intron. It may screw up splicing in various ways. So I don't know how successful they're going to be in the long run with their whole genome project. However, lucky for us, they had, when I hit their website, I noticed that they had generated 5 gene traps in 5 rest alleles, 5 different lines. And so I wrote them and asked if they would collaborate with us to sort of do the proof of principle for them in vivo. So they published a PNAS paper where they tested this in cell lines, and it worked really well. But it hadn't been tested in vivo, so we decided to test it with rest. And I'm just going to go very quickly through the data because it's a little bit preliminary. So we have we've been testing 2 gene trap lines. This is the exonic structure of rest, and this is where the 2 insertions are. And then here are the kind of things we need to know. Is the homozygote gene trap really a knockout? It better be. Okay, so you have a gene trap that's non-functional. That's a het. If you make a homozygote, it should be an embryonic lethal. So we had to test that. On the other hand, you want to make sure that the flipped version is wild type, because then you want to flip it again with Cre. And it turns out it is. As far as we can tell, mice that contain the double inverted are perfectly wild type. So then that allows us to do a lot of different experiments. One experiment we can do is just look to see, using LAC-Z, where rest is expressed, for example, in the adult. So one question that's been outstanding is whether rest plays a role in the adult nervous system. And that's been very difficult to answer because the antibodies don't work too well when you don't have a lot of rest. And I'll show you a little bit of data of that. And then, of course, we want to cross the lines with different Cres. And the one that we've been focusing on is a nestin Cre, where we can knock rest out specifically in the progenitors. So in the last 2 minutes, I just show you that the system works. So here's the K-out, the knockout of rest in the homozygous, and you see we do in fact get rid of rest protein and rest RNA. The knockout that we generate with the gene trap allele looks exactly like the knockout that David Anderson generated in 1998. The embryos are small, and they seem to have some kind of funny heart phenotype. They died around E10.5, and I can't tell you why. I don't really know. Using the LAC-Z from the gene trap has been really nice. So this is a picture of an adult mouse brain. This is wild type. This is our gene trap. And we were very surprised to see rest in only one place in the adult brain. And it's in the place that you might expect to see adult neural stem cells. And we've been able to confirm that the LAC-Z expression actually does reflect promoter activity from rest. So this is an immunohistochemistry picture of rest showing that we can indeed detect endogenous rest in the ependymal layer surrounding the third ventricle. This again is in adult mice, and also in the subependymal layer where you expect to see neural stem cells. So we're actively trying to figure out what the role of rest might be in the adult stem cells. Does it orchestrate the same kind of chromatin structure in an adult neural stem cell that it does in an embryonic stem cell. And finally, last slide is just to say that if you get rid of rest at the nestin-positive progenitor stage, you do in fact have a very robust phenotype. And the phenotype is that the brains of these mice are really disorganized around the region of the corpus callosum. So actually, when we were doing our first dissections of these mice, we noticed that the two halves of the brain just kind of fell apart into two halves. Your brains are normally connected, of course. And when we did the dissection, the two halves just kind of fell apart. And it turns out that that's because these mice are missing the corpus callosum, which is a big bundle of axons which keeps the two halves of your brain together. And in addition to lacking this central structure, there's a lot of disorganization around the hippocampus and in the cortex and so forth. So I don't think rest plays much of a role in ES cells, ES cell to neural progenitor, but I think it's absolutely critical in getting a mature neuron from a neural progenitor cell. And so I just thank the people whose work I talked about. I already mentioned John Yates, the postdoc we've been working with, who's a very talented person, Lu-Jin Liao, actually, who's looking for a job, and I think he's just terrific. And Stu Orkin for the help with the ES cells. And Tamila Nechaporak was the person who's been doing all the work with the gene trap mice, and that's actually been in collaboration with this GGTC consortium. And whoops, I see Narit Ballas has been eliminated. She now has her own lab, but she did almost all of the early work on embryonic stem cells and rest. Thank you.
We have time for a couple of questions.
Yeah.
You mentioned, the issue with rest knockout in ES cells because of this drive for self-renewal, but you didn't mention any phenotype differentiated cells. So, for example, if you knock out rest in a fibroblast,
There's no effect because you don't de-repress the genes there.
Yeah.
So in ES cells, you get very robust de-repression of a lot of genes, but by any criteria, we just don't see any difference in ES cell biology. We haven't done chimeric, transplant experiments or anything of that nature because there was nothing in tissue culture that led us to suspect there was something wrong. Yeah.
In your comparison of the ES cells to the fibroblasts, and you were seeing DNA methylation in the fibroblasts, but not the ES cells. But was that DNA methylation in the CPG island? Or
No, there's a CPG in the rest binding site, and that's methylated in fibroblasts. And then on either side, I wouldn't call them islands, but there are CPGs that are methylated.
So it's on sort of the edges.
Yeah.
And then you looked for when any CPG gets recruited there, is it
okay.
itself to the methylation, or it's just interacting with the co-repressor?
We were never able to co-IP rest with MECP2, and MECP2 did not turn up in our mass spec at all. I think it's a different complex. So I don't know how I'm assuming it's binding to methylated CPGs, but I don't know. But it doesn't seem to be physically part of rest complex. Yeah.
When you knocked down rest and saw the loss of the trimethylation of K27 on the target genes, did you look at non-rest target genes to see if you were getting a global loss of K27 or if it was specifically just to that set of target genes?
We did look at one other gene. No, we didn't. No.
That would be interesting.
Yeah, that's a good idea.
The whole genome or just that one category.
Yeah, that would be good. Yeah.
I was curious if anybody done any looking for an E3 ligase that might have been upregulated in the progenitor cells relative to the embryonic stem cells to indicate why it's suddenly being chewed up.
Mm-hmm. So there are two Nature papers that were published, one by Michelle Pagano and one by Steve Elledge. And both of them identified an E3 ligase and an F-box protein, CUL1, and a portion on rest, which they felt was being targeted for the proteasome pathway. Most of that work from those two labs was done in HEK cells, where we don't see a whole lot of turnover. So they gave us their mutated version of REST that was missing a phosphorylation site that they said was important for this targeting. And we don't find it has any activity. It doesn't stop stabilization in progenitors. So I think it's a completely different mechanism in progenitors. And our latest work kind of suggests that it's through GSK beta. So.
Great, thank you, Gail.